So first, I want to thank Bruce and the organizers for inviting me. It's been years since I've been back in Boston, and it's absolutely amazing. I really welcome discussions, feedback. My research program is very nascent, as well as the field that I'm working in. So I'm really excited for those discussions. So just by way of introduction, my lab studies ion channels that are targeted by GPCRs, and we try to identify novel interactions and then ultimately evaluate how their interactions influence action potential firing. And I took interest in these serotonergic neurons that live in the dorsal raphe nucleus. These cells are not autonomous pacemakers like the dopamine neurons, but they require sub-threshold drive, synaptic input. So they're considered conditional pacemakers. And what's unique about them is that they require tonic noradrenergic input that activates these alpha-1 adrenergic receptors, which are GPCRs. And that's what provides the sub-threshold drive in vivo for firing. Once they reach that, then they have beautiful clock pacemake firing. So here was the first example in the early 80s from George Agajanian's group. They're recording in vivo an anesthetized animal. Oops, I backed up. And here you're just seeing a raster plot of action potentials. They locally apply an antagonist for the alpha-1 receptor. The cell becomes quiescent. You can chase it off by applying noradrenaline, and then eventually that antagonist washes off. So in moving from an in vivo preparation to in slices, the cell bodies that are providing that noradrenergic input are severed, but you can still stimulate and evoke noradrenaline release, and you get this really beautiful synaptic current. So all of the work you're going to see today is in mouse brain slices from dorsal raphe serotonin neurons. And here's an example here where we stimulate that evokes noradrenaline release, and it produces this very long-lasting, slow EPSC. If you throw it in current clamp mode, you can see it really robustly causes action potential firing. And this is through activation of the alpha-1 adrenergic receptor. If you antagonize it with prazosin, that eliminates it. And this has been known really since the early 80s that this was the requirement. And we know that serotonin neurons and serotonin release is a huge pharmacological target. There's a number of conditions that involve serotonin. And so I came into this project really interested in the fact that we'd known this for decades, but the ion channel that was responsible for this current hadn't been identified. So that's what I sought out to do. And the leading theory when I came into it was that it was closure of a potassium channel. So the first thing that I did was I just designed this really simple voltage ramp, taking advantage of the fact that this current is nearly 30 seconds long. I can ramp before, I can stimulate and ramp again and do subtraction. And right away, it became very obvious. I'm going to keep doing that. It became very obvious that that synaptic current was not reversing anywhere near EK. Okay. Similarly, if I increase extracellular potassium, our holding potential is minus 65. And so at 10.5 external, that's right about where we're holding. We never saw a decrement in amplitude of this synaptic current. So it started to look like we're probably not looking at closure of a potassium channel. So then I just extended our voltage ramps, a little bit of different protocol, but again, taking advantage of the fact that that synaptic current is so slow that we can really ramp through during the current and this is a single cell, and you can see the subtraction here reversing around minus 30 millivolts. So that told us that our channel has mixed permeability. Primarily, it's carried by sodium entry. And so in this experiment, I replaced sodium with NMDG and then applied noradrenaline. Here, that's our exogenous agonist. And you can see in NMDG, that current is completely gone. So we have a channel that's non-selective. It's mostly carrying sodium in. And this led us to think maybe one of our candidate channels was this non-selective sodium leak Nalcan current. And this Nalcan current, I'm showing you an example from Dejan Ren's group where they expressed it in cells, and you're seeing the current here. It's really nicely blocked by the trivalent gadolinium at 10 micromolar, and it very poorly washes out. So we tested gadolinium on our synaptic current, and it has no effect. Okay, so continuing on with basic biophysical properties, what I'd already noted to you was that this synaptic current is very noisy. Has this basal noise, and then at the peak of that EPSC. And so we did noise analysis, and it indicated a consistent conductance state about -1 picoamp. There's some features about the noise analysis that if anyone really loves this, I'd love to sit down and we can chat more about it. For the context here, what we were most found promising was that it was likely one type of channel. So that kind of helped so that we might be able to identify it. It was also rectifying. And so I created this voltage protocol where we're holding at -65, and then we give a pre-pulse conditioning stimulus, either from -120 all the way up to 30, and then step back down to -120. And this is a result, this is just an example of that exact thing before. So in control, and then after noradrenaline application, you can see that really nice inward current, and then we run through our voltage protocol. Here's an example right here of one at -65, a step to 30, and then back down under basal conditions, and then after noradrenaline. So the gray here is control, and then black here is noradrenaline. This is an example of the noise. Can calculate conductance from this, and what we saw was that it's decreasing right around the reversal potential. So what it's saying is that it's preferentially passing inward current, and as we depolarize, it's losing the ability to conduct, which I think you can actually even just appreciate right here, where it seems like it's starting to close and then potentially reopening. Okay, so we generate a whole list of biophysical properties, and then the next thing was to probe channel identity with selective toxins, or these gifts from nature. And at the time, one of them really drew interest from other work that had been done in the field. And that was a synthetic version of the Jora spider toxin, which you're seeing right here. And that is NASPAM. So this is that synthetic version. And as soon as we applied NASPAM, it really effectively blocked the current. And it takes some time to wash out, but it was able to recover. What was interesting about this is that NASPAM is known widely as a poor blocker for the calcium permeable ionotropic glutamate receptors. I'm showing a structure here of NASPAM fitting in that central cavity and then down into the selectivity filter. It is known to be an open channel blocker of these calcium permeable IGLURs. So I was doing the recordings in the presence of NMDA, AMPA, and kainate blockers. And so then I had to step back and ask myself about the family of ionotropic glutamate receptors. So here you're seeing just a sequence alignment here, homology, shared identity. And up here, we're starting with delta. So you have your two variants, grid 1 and grid 2. These are your genes encoding GLUT1 and GLUT2. Your AMPA, kainate, and then the NMDA receptors down here. So with the drugs that I was already using in our ACSF, we could eliminate your AMPA, your kainates, and then any of the NR2 containing NMDA receptors. What was left were the orphan delta receptors, and then the NR1, NR3 subunits of the NMDA receptor. So there was some more pharmacological work that went into deciding which one to target, but we knew we needed a genetic strategy. And so we decided to choose GLUT1 and go in with a CRISPR-Cas9 strategy to functionally delete it from these neurons. And lo and behold, it completely eliminates the current. And so this was a viral strategy. And so we had the advantage of having some cells in the slice right nearby that weren't transduced, and they had a synaptic current. Those that were, it was gone. So we're building a case. We've got this pharmacological and a genetic strategy to indicate that this synaptic current is carried by the delta 1 glutamate receptor. So what are they? I told you they're a member of the ionotropic glutamate receptor family. They're found in nearly every region of at least the mouse brain. So you're starting up here. This is mRNA intensity at the olfactory bulb all the way down to the cerebellum. GLUT1 is in red and GLUT2 is in black. And I think you can appreciate, even if you can't read these individual regions that it's in pretty much every area. I'll just note here the Purkinje cell layer. This is GLUT2, where it's very highly enriched, almost 20-fold. That's originally where GLUT2 was first established with a spontaneous mutation in the 60s that generated the lurcher animals. And so that's a constitutively open channel. Most of what we know about Delta is from lurcher. The other things to know is that they do not bind glutamate, nor is there a known gating ligand yet. They have an overall similar structure to the other IGLURs. They're tetramers. They have this Y-shaped structure. Here's your amino terminal domain, the ligand binding domain, and then the pore region. They have that traditional clamshell structure. There are some features about them that are a little different from mammals that are more a little bit similar to plant-like receptors. But I think the key feature and it's a key limitation that we face right now is that expression systems of native receptors have largely failed. The exception to that are these constitutively open mutants where there's mutations in the pore. And there's a lot of theories as to why expression systems aren't working yet. But I think that once we overcome that obstacle, then we can really start to push this forward. Okay, so I told you that GLUT1 current in brain slices seems to be stimulated by noradrenaline. I just want to make that very clear that it is not noradrenaline acting on the channel, but that is requiring activation of these alpha-1 adrenergic receptors and signaling through a yet to be identified pathway. What we also found in brain slices was that it appeared that GLUT1 was carrying this tonic current. And so you're seeing a similar experiment here where I've applied NASPAM. It's just a compressed time scale. This is that alpha-1 EPSC we're evoking every 90 seconds. Here's bath application of NASPAM, and you observe this apparent outward current and reduction in membrane noise that comes back down as the alpha-1 EPSC starts to recover. This current that we observe with NASPAM is eliminated if we go to our low sodium solution with NMDG, and it's also gone in the CRISPR-Cas9 null animals. So what we think is happening is that these channels appear to be open some level at rest, and they're allowing just a tonic influx of sodium. Now, at the time, we were using very high concentrations of NASPAM. This is 100 micromolar. That's not uncommon in the calcium permeable AMPA receptor literature. But as we know, all drugs have off-target effects if you use too high of concentrations. And so I had one of my graduate students, Drew, who's here at the meeting. He's working on generating a concentration response curve to NASPAM and to another spermine analog, pentamidine. And so you're seeing the current here. This is just the remaining synaptic current by concentration. And we're getting IC50s in the low micromolar range, which really aligns nicely with what's known at GLUT2 lurcher. So, we were definitely using much higher concentrations than we needed in earlier experiments. Okay, so outstanding questions. This is kind of what keeps the lab up at night, or at least keeps me up at night, is how does activation of alpha-1 produce this current? What allows for tonic current, and is it really just a product of GPCR signaling? And so we've known for a couple of years that alpha-1 GLUT1 current. So I'm going to now, instead of referring to it as synaptic current, just to distinguish it from tonic, I'll refer to it as alpha-1 GLUT1. It's dependent on GTP-GDP exchange. So if we record with GDP beta S in the internal solution, right after break-in, it runs down very quickly. In contrast, the lithium control internal is very stable. If you want to learn more about the GPCR pathway, I refer you to Drew's poster. It's number 12. I don't think his abstract made it into the book, but please stop by. I know he's more than happy to talk to all of you about it. So in contrast to the alpha-1 GLUT1 current, though, GDP beta has no effect on this tonic GLUT1 current. And so this is Daniel and Aaliyah. They're both post-bacs in my lab, and Aaliyah is now a graduate student. They took on this project. So they're patching with GDP beta S. They're applying noradrenaline, really beautiful inward current that's gone by the second application. And then they assay for tonic current by applying NASPAM, and you can see it's really nice here. There was never any change in amplitude between our GTP, our GDP beta S, or our lithium internal. So that led us to conclude really that tonic current and this alpha-1 glutamate current are arising from different mechanisms. And so you can have kind of two schools of thought, two teams in the lab. I oscillate between both of them or all of them at the same time. One is that glutamate is gated by a ligand, and we just haven't identified it yet. And maybe in our brain slices, it's there and it's ambient. The other side of that is that glutamate might have just a stable apo state. And so in the absence of ligand, somehow it flickers open, or when it does enter an open state, it stays there for enough time to generate current. I was really interested too in the intersection, which I know there's a lot of other possibilities here to consider. But what about they're being bound, but they're desensitized? They're in their very low conductance state. And there's evidence for this in the IGLUR literature that even desensitized AMPA receptors can produce a steady state or a tonic current. So this was work by Stuart Kulkandy's group, where they were looking at AMPA receptor currents expressed in cells. Here, cyclothiazide is going to block desensitization, really nice current. And then they used mutants that allowed these receptors to desensitize more. And what they observed was this steady state current. And so as you enhance desensitization, you get more of this steady state or more tonic current. So that directed our attention to what's known about desensitization at the deltas. And we turned to work done by Casper Hansen when he was with Steve Ternellis. And they had found that extracellular calcium modulates desensitization of GluT2 Lurcher. And there was a site that stabilizes the ligand binding dimer interface. And when calcium is there, it stabilizes them so they don't desensitize. So the really simple logic, more desensitization, more tonic current. Maybe if we went to calcium free, we could break this and we'd get more desensitization. Do we observe more tonic current? So we did these experiments in nominally calcium-free external. We're using that synaptic current as a control that we've washed out enough calcium. And then we assay for tonic current. Here's NASPM. And lo and behold, it's just a little bit bigger. And then we thought, well, what about higher concentrations of calcium? So we increased it to 2.4 or 4.8 external millimolar, and now we saw a reduction in that tonic current to the point where it's almost getting really difficult to distinguish from membrane noise. So then returning back to the alpha-1 GluT1 current, we also noticed that this was modulated in two different ways by extracellular calcium. So in the first example, I'm showing you, this is the alpha-1 GluT1 current in 1.2 calcium. That's our standard recording solution. And then we'll switch to elevated, either 2.4 or 4.8. We switched to 4.8 millimolar. We observed a nice reduction in that current. This is an example trace of that. So in control, 1.2 and then 4.8. So you're getting about a reduction of about almost half. And then if you scale their peaks, this tau, this decay here, is a lot slower in 4.8. And so if we were thinking about elevated calcium stabilizing a dimer interface, maybe it's prolonged because it's not able to desensitize. It's just one theory. But this reduction here led us to the question of maybe calcium is just blocking the pore. And so we turned to a page from Bruce Bean's work of looking at MK801 at the NMDA receptor and how it competes with magnesium. And so MK801, of course, is a use-dependent blocker. It's open channel blocker, nicely illustrated here, where you activate with MK1 initially, get a current, and then it's blocked. And it's really long lasting. If you look at recovery of that response later, you can see it's very small. But when you have magnesium on board, so here first looking at low magnesium conditions, here's MK801, and now we're focusing on the recovery. Without magnesium, recovery is very poor. But if you apply MK801 in the presence of high magnesium, then recovery is a lot faster, it's a lot better. And so the conclusion there was that that magnesium is somehow protecting the channel from block by MK801. We did the exact same experiment now looking at calcium on GLUT1 current. We found, so I'll walk you through this experiment. So here we're recording in 1.2. This is the alpha-1 GLUT1 current. We switch to 4.8, we get a reduction. And then we apply NASPAM. And right away you might observe we don't achieve as good of block in high calcium. We start to wash it out in 4.8, and then we switch rapidly back to 1.2. This is the example here. So in these gray circles is in 1.2 calcium. NASPAM block is almost complete, and then it's slow to recover. You've seen this before. This in the open circles is 4.8, so we get a reduction. Here's NASPAM coming on, and then right at this point, we switch back to 1.2 and observe this accelerated recovery. And so our best interpretation is that calcium, just like magnesium, is protecting the pore from block by NASPAM. So after we reported this, putative block by calcium, there's a really nice paper from Steve Ternellis's group looking at the constitutively open version of GLUT1. So that's here in comparison to Lurcher. And they observed this really striking difference where calcium appears to potentiate the GLUT2 Lurcher, but it's inhibiting GLUT1, at least the constitutively open. So that might be a nice distinguishing feature between the two. Okay, so I just want to go back to this idea because I prefaced this is that we were getting into this question because we wanted to know if tonic current is from desensitized low conducting receptors. And this result may be possible, that's possible. Still, we can't formally rule it out, but we also can't rule out the idea that there might just be block of these channels at physiological calcium and that we're alleviating that when we're switching to calcium free. Okay, so I'll just conclude with a few take-home messages here. GLUT1 is absolutely a functional ion channel in brain slices. Presumably everything is intact that it needs in an intact system. Maybe that's why we've had success when other systems have failed, or if there is the expression of accessory subunits, or if there is an ambient ligand, everything is there, presumably that it needs. We have two different forms. We have this G protein independent tonic current, and then we have a G protein dependent alpha-1 GLUT1 current. And we've found that it's really well modulated by extracellular calcium, likely in at least two different ways. So I just want to thank the lab. Hopefully I've mentioned them as we've gone along. I've had an incredible group of students, graduate students, undergrads, and postbacks. And then of course, funding that has enabled this. And I'm happy to answer any questions.
Maybe I can start, Stefan. I'm curious about other GPCRs and in other, like muscarinic ACH receptors. It's always been curious what conductants they interact with.
Right. Yeah. So what I can say is that other groups before we entered this looked at orexin and histamine and phenylephrine in dorsal raphe serotonin neurons. They argued that there's convergence on the same channel. So that would say that maybe that's true. We've tested mGluR in this system and we get mixed results, a couple different channels. But others say that mGluR and the dopamine neurons might signal to GLUT1. So I think it's a completely open question. And we really just need someone who's interested in testing all those possibilities. Yeah. Yeah.
Well,
Thank you. I'm sorry. I'm trying to think of some old literature I'd read about GLUT1 receptor and the role of cerebellin. And how does that story figure in here?
Yeah. No, thanks for.
And also, isn't when cerebellin's present, doesn't it have endogenous agonists, glycine and
Mhm.
D-serine, if I remember right.
Yeah, so no,
okay.
I'm really glad that you're bringing this up because in the interest of time, we have to like kind of triage the whole story. But the idea with these receptors is that their Y structure is quite floppy compared to the other IGLURs. And so folks have shown that they bind presynaptic orexin and soluble cerebellin, and that forms a transsynaptic complex that might confer the mechanical stability to gate the channel. Now, in regards to D-serine and glycine binding, they do, and they bind really well. They don't seem to gate the channel. What they've known as a ligand is that they, like the sort of non-ionotropic functions of NMDA receptors, binding of D-serine can somehow change its function as a scaffold protein involving the C-terminus, not the pore region. So in our hands, D-serine and glycine acts more like an antagonist. And that's true also for those that have studied literature. The idea is that maybe it's shifted it to a desensitized state too fast without traveling through an open state. But yeah, I mean, so calling it an orphan, I think is probably not quite correct because we do know all these factors that bind, but nothing yet has directly opened the pore. Yeah.
Well, no further questions now. We
Yeah,
can.
one more.
Thank you. Sorry. Have you tried the like the voltage clamp method, like single ion channel analysis for voltage clamping method?
Have I tried what?
The voltage clamping instead of like current clamping.
I think there's like a strange echo in your ceiling that I'm not. Does someone?
Uh,
Yeah. Oh, voltage clamp versus current clamp. Yeah. Yeah. I mean, so we do both. We do both routinely. This really, yeah. So I guess even with the tonic current, we see a reduction in the resting membrane potential, exactly what you'd expect for Ohm's law. So yeah, we can go between the two. Mhm.
Well, we can
All right,
continue
thank you.
more over lunch.